Justin. It's now my privilege to welcome to the stage our keynote speaker, Clinical Associate Professor Michael Buckland. He is a Senior Staff Specialist and the Head of Department of Neuropathology at Royal Prince Alfred Hospital in Sydney. He is the founder and director of the Australian Sports Brain Bank, co-director of Multiple Sclerosis Australia Brain Bank, and head of the Molecular Neuropathology program at the Brain and Mind Centre. This evening he will talk to us about his research from a molecular level. Ladies and gentlemen, please make welcome Michael Buckland.
Thanks very much, and thanks for the kind introduction and for the invite for being here. I've got to say, as a neuropathologist, I occupy a very privileged position in New South Wales Health, in that I'm a pathologist that spends all my time looking at brain and spinal cord tissue, both under the microscope and increasingly using molecular techniques. And brain tumours have been my first love and still my primary love, although I am now distracted by a few other things like the Sports Brain Bank and the MS Brain Bank. But in fact, the fundamental principles amongst all brain disease sort of remain sort of the same. And I also want to thank Michael Back and Helen Wheeler. I've known them since I started in this role. They've been some of the best collaborators I've had over a long period of time. They've supported my work, and hopefully I've supported their work as well. So I'm really pleased to be able to give this address today to the Brain Cancer Group. So that's the title of my talk. So I'm just going to take you on a little bit of a historical journey. So really, for almost 100 years, the classification of brain tumours has been based primarily, exclusively, on how they look under the microscope. So we get these big names like astrocytoma, oligodendroglioma, because the astrocytomas, they look like normal brain astrocytes, which are the cells that support the nerve cells in the brain. And oligodendrogliomas look like oligodendrocytes, which are the cells that produce the myelin that coats the nerve cell fibres. And it's that myelin that breaks down that causes multiple sclerosis. So for almost 100 years, just how things looked under the microscope. And then in 2016, the WHO released a revised classification that for the first time included a layer of molecular information as well as the microscopic appearance to reach a final integrated brain tumour diagnosis. The idea is something a bit like Google Maps. You know, if you go on Google Maps, you can either look at satellite view or street view, and there's these layers of information you can look at, and it's similar with the brain tumours. You want layers of information, and all those layers then collapse down to give you a final diagnosis of what the tumour is and how aggressive, predicting how aggressively it should behave and how it should respond to treatment. So this is basically how all our reports come out. We first say how it looks like under the microscope, we then say what the relevant molecular alterations are, and then we issue a final diagnosis integrating all those key components. Last year, the WHO released their 5th edition of the classification of tumours of the central nervous system. It has gone in even harder on the molecular classification. It still retained the histological classification. What it looks like under the microscope is still the first step in that process. But there's now so much more molecular information that's required to give you your precise diagnosis. There's over a dozen new entities based on the fact that things that used to look the same under the microscope, now actually there's something that was once called an ependymoma. Now there's 6 different types of them, because we now understand that even though they look the same, they have different genetic lesions and hence they behave differently. And for the first time as well, there's been an emphasis on tumour location. We now know that say an astrocytoma of the cerebral hemispheres, say the frontal lobe, actually is not the same as an astrocytoma of the cerebellum. even though they look the same, the location also dictates what sort of molecular lesions they have in them and how they're going to behave. And so not only the molecular now, but now they've also introduced location as a very important component of how we classify the tumours. I just took this photo today from just from my desk. So we've got two books there. The bottom book was the WHO classification, the 4th edition, and the top book is the WHO classification, the 5th edition. So this is, it's more than twice as thick. It's a bit overwhelming at times, but what it really is reflecting is the amount of new information that has come out over the last 10 or so years. Unfortunately, I think brain tumours have lagged behind things like breast cancer, colon cancer, but that they're catching up fast, which is wonderful to see. That's the table of contents, which essentially is the classification of brain tumours. There's quite a lot there. It's expanded enormously. One other thing that, and I'm going to talk about a couple of these tumours, but one of the very important fundamental things is the acknowledgement now that childhood brain tumours are actually completely different beasts to adult brain tumours. In the past, they were lumped together, but now it's realised that, in fact, developmentally, molecularly, they're very different beasts. So this is how we actually classify tumours today in 2022. And we start off at the top with the histology, or the microscopic appearance. And then you'll see down the left-hand side, oops, sorry, I'm trying to get that, oh, I got it. I'll use this. You'll see, down the left-hand side here, these are the key molecular features we now need to assess, and in fact it's now requisite, we need to assess them to be able to arrive down the bottom to that final integrated diagnosis. And I'm just going to talk about a couple of those. The first one is IDH. So IDH mutations appear to be very fundamental mutations. They're the earliest known mutations that occur in a group of brain tumours that predominantly affect adolescents and young adults. When I say young, up to the age of 54 or so. I'm 54, so I guess that number might change, but in fact it is actually 54, the number that's listed. And they seem to be the earliest alteration we know of, and a very stable alteration. It doesn't matter what you do to the tumour, you cut it out, you blast it with radiation, it's always there. So very, very important, and really the first step of that molecular classification. There are now some clinical trials of IDH inhibitors that are showing promising results in sort of combined phase 1, phase 2 trials, but they still, I think they're still a long way away from the clinic. We're looking probably at 10 years or so. The next lesion I want to talk about are these two genes, CDKN2A and CDKN2B. And it seems in the group of IDH mutated tumours, they actually are very important predictors of behaviour. And I think we're going to hear more about that from our next speaker, which I'm really looking forward to. And so what's another change that's happened now is that we used to say this tumour is a grade 2, this tumour is a grade 4, based exclusively what it looked like under the microscope, even with that first layer of molecular information. And for now, in the 2021 classification, we realise that even if the tumour looks like grade 2 to us under the microscope, if it has these deletions, we know it's going to behave like a grade 4. So now the molecular information is starting to almost override the microscopic appearance and tell us about behaviour that we would not have recognised in the past. And on the similar note, these changes here, we're talking about TERT promoter mutation, EGFR amplification, and chromosome 7 and 10 changes. We have to assess all these because in the most common and most deadly brain tumour, the glioblastoma, again, these alterations tell us this tumour is going to behave badly, even if it only looks like a lower grade tumour under the microscope. So they become very important parts of the assessment. So even if we think, oh, this is a grade 2 astrocytoma under the microscope, if it has these alterations, then we know it's going to behave like a glioblastoma. So important in brain tumours compared to a lot of other organs, because it's pretty hard to get to the brain tumours, and generally, for some reason, neurosurgeons don't like to sort of scoop out a large amount. They want to give us the smallest amount of possible to make a diagnosis. And so what that means is that they may not actually sample the worst looking bit. And if we can use these molecular alterations, they don't have to sample the worst looking bit to be able to get an accurate diagnosis to inform appropriate treatment. So at RPA Neuropathology, we're very lucky to have a dedicated glioma gene panel. We use an iron-tolerant deep sequencer. There's certainly other sequencers out there. We're able to cover alterations of all these genes on a single sequencing run. So in the past, you'd have to sequence this gene and then think, oh, actually, now we're going to have to sequence this one and this one. Now you can do them all simultaneously, which means you can, but one, save on tissue, and two, save on time, because you get all that information within clinically relevant time frames. And the beauty of these machines is they require very small amount of input material. So 20 nanograms of DNA, so that's 10 to the minus 9 grams of DNA is required. Very small amount and very suitable for small biopsies. When we got the panel up and working, we did an audit of our first 4 or 5 months of sequencing to look at what the tumour was diagnosed histologically and then what the final diagnosis was after we'd run it through the sequencer. And in fact, just over 20% of cases, their diagnosis changed once you ran them through the sequencer. So it actually does have quite significant real-world impact getting this molecular information. This is Grace, our molecular scientist on the left-hand side, who's looking a bit more frazzled than she used to, because you can see the increased number of cases as more and more oncologists and surgeons realise the importance of getting this information quickly. And we're expecting those numbers to keep going up. And this is just our numbers, which is not only the gene panel sequencing, but individual gene testing as well. And they're also showing that same curve. And we just sort of hit 1,600 last year, probably going to break 2,000 this year. So it's turned out to be, we've been in the right place at the right time with the right support from Michael Back and colleagues to be able to roll out this service at the right time. This is one example that came through a couple of years ago. This was a 57-year-old female that had what looked like a diffuse glioma in her motor strip. So in the part of the brain that controlled the motor functions of one part of her body. And the bad bit looked entirely restricted to that motor strip. And so it was a real dilemma. In the past, the surgeons would, if they wanted to get the right diagnosis and the right grade, they'd have to go into that motor strip with the real risk of causing significant paralysis or motor deficits. But now we realise that, in fact, we don't, as I said before, we don't need that worst looking bit. You can biopsy the periphery of it, and then you get the molecular information required to give a final diagnosis. So in this case, it turned out to be an anaplastic astrocytoma, which is a grade 3 tumour. But then once we'd done the sequencing, it had the molecular signature of a glioblastoma. So we could get to that diagnosis without damaging the patient. I'm just going to use one more example of the new classification of a certain type of brain tumours called ependymomas. They're not that common, but certainly when I was a younger man, there were 3 types of ependymomas, and everyone knew what they were. But now, unfortunately, there's 9 types based on the molecular classification. And I know this is a very busy diagram, but I just want you maybe first of all focus here. They're broken up into tumour location. As I said before, it now seems that even though they look the same, they're probably derived from different precursor cells that reside in different areas of the brain and spinal cord. So they're divided up by location, as well then by a large number of different molecular alterations. And the way that this has been discovered and defined, which has really happened in the last 4 or 5 years, is by a new technique called DNA methylation profiling. This is a methylation array, and it's called 850K because there's 850,000 little dots on there, unique areas to interrogate the methylation pattern of the tumour. So methylation is something that sits on top of the DNA. So if you sequence the DNA, you don't see it, but it's a chemical modification that sits on top of the DNA, which is very important in either making the DNA be active or be silent. And a collaboration of a large number of German institutions has sequenced done this array testing on several thousand of different brain tumours and found that there are very distinct clusters, and you see all these dots over here on the right-hand side. These are all distinct clusters of methylation patterns. So you can't see it here, but say this little blue blob here, that might consist of 20 or 30 different individual blue dots that are tiny, but they're all clustering together so they look like a big blue blob. But it seems that a specific tumour type will cluster together on this methylation array. And if I show you down here, these are now all the ependymoma types. So what we thought was just a couple of different, or 3 different types, now you can see how distinctly they actually declare themselves when you look at them through the methylation array. And this is something that is certainly going to be very important in the next 3 or 5 years of tumour classification, brain tumour classification here in Australia and elsewhere. And the reason why it's important, so if you imagine if you had an ependymoma clinical trial 10 years ago, and they said, okay, we're going to get everyone with an ependymoma and put them in, we're going to give them this drug X. Now we know that, in fact, that's not a homogeneous group you're testing. It's likely that there's probably 9 different tumours mixed into what you think is a homogeneous group. So when you then give them the drug, it might be that some people with a specific tumour type do well, some people with a different ependymoma type no benefit, and some do worse. But because you think they're all the same, you aggregate all those outcomes and then you say, this drug's no good. It's made no difference to outcome. So the stratified medicine approach is by using these markers, we can say, all right, so now we're going to test people with only this subtype of ependymoma, and maybe we'll find a drug that works. Or we'll test everyone with an ependymoma, but then we'll break down the outcomes based on the tumour type that they have. So this is a mechanism that hopefully will accelerate sort of clinical trials in the very near future. And then I think as we've already heard, that precision medicine is the ultimate goal, where based on the molecular profile, we're able to individually personalise someone's treatment based on the druggable targets that they have. I think that there are a couple of examples of that just starting to come into the brain tumour world, and this is probably the best one, which is BRAF targeted therapy. We know BRAF is commonly mutated in melanomas, and these targeted therapies to attack that BRAF mutation have been responsible for a lot of the good outcomes we've heard of in melanoma in the last 5 or 10 years. Unfortunately, in brain tumours, it's a really, really uncommon mutation. So this is not going to benefit many people, but it is a fantastic proof of principle that if you give that targeted therapy, precision medicine can work in brain tumours. We just need the right drugs. And that's something that's certainly ongoing. So I just wanted to finish off with a little bit of our research, and some of this we're collaborating with the Brain Cancer Group, which is fantastic, and as I said, they're very collaborative. One piece of research is a blood test we're developing to monitor brain tumour activity, assessing little circulating particles in the blood called exosomes, and this is from an original proof of principle paper, and this is a heat map. We've got healthy cases over here versus people with a glioblastoma over here. And you can see some genes are upregulated when they're red compared to some genes downregulated when they're blue. And by using this combination of assessing circulating genes in the exosomes or these particles, we're able to distinguish someone that's healthy versus someone with glioblastoma. And we certainly hope that in the future we'll be able to monitor tumour activity much more rapidly in real time, not necessarily needing an MRI scan, but through a blood test. And the other big research project we have is the Sydney Brain Tumour Bank and the GliaNet study. And this is driven by Kim Alexander, who's a good colleague of mine. And that's her in the background. She's a bit out of focus there, I think. And this is a longitudinal collection of brain tumour and blood specimens. So anyone that has a brain tumour cut out that signs up, we're able to bank that tumour and use it for research, as well as take serial blood specimens to further our blood test research as well. I know Kim's been talking to Michael and Helen Wheeler and others about rolling this out at North Shore as well, at least so we have the same data sets, so the databases will talk to each other. So if someone's looking for 100 oligodendrogliomas, we can talk, say, to the North Shore Tumour Bank and know that they will pull out the same sort of tumours that we're pulling out. So it's very important work as well. So this is the team, and I think this is Grace here, who's our key molecular scientist. This is Maggie, who really runs the lab, which is fantastic. And a slightly informal shot of my other pathologists that work in the group. We're lucky that we've grown. Dr. Lavinya Satgunaseelan does a large amount of the molecular work now. Joanne Tsai is an excellent diagnostician. And just to bring in the North Shore thing again, we're now lucky to have Dr. Wei Chu, who works mostly at North Shore. She's come to join us for 1 or 2 days a week, which is terrific. So thank you very much. It's been a quick tour through some of the baffling molecular stuff, but if we've got time for questions, I'm happy to take any.